All right, what's going on YouTube? It's Attorney, it's our tutor, and today we're going to be talking about levels of protein structure. So first off is going to be primary structure. So basically primary structure is made up of peptide bonds, and these are covalent interactions between the amino group and the carboxyl group of two adjacent amino acids. Now, so we'll write this down. Oh, whoops, we're on the wrong marker. So this is a peptide bond, and these form, it's a dehydration reaction, which means water is displaced in the process, and I've highlighted the peptide bond in red, as you can see. So it's near the chiral carbon of the amino acid. But anyways, let's get into the secondary structure. This is just a brief overview. So I can go more in depth if needed in a future video. So the secondary structure, so it's going to be intramolecular hydrogen bonds, and the two big structures is going to be the alpha sheet, or the alpha helix, sorry, and the beta pleated sheet. And a fun fact about these is the amino acid proline, due to its rigid structure, is able to break alpha alpha helixes and beta pleated sheets. So typically at like the end of an alpha helix, you're going to have a proline to kind of break that interaction or break that secondary structure. But anyways, let's get into tertiary structure. So tertiary structure is going to be the hydrophobic interactions. So this is going to be like your nonpolar interactions, and a big one is going to be the disulfide bridge. So this is a huge one in biochemistry, and you might see it in cell biology, but the disulfide bridge is going to be between two cysteine residues. So this is an amino acid that has sulfides at the end. So I'm not going to draw the whole thing, but this is the interaction. So this is the R group of the um, of the cysteine molecule. So it's going to undergo oxidation. And when it undergoes oxidation, you get this disulfide bridge, which is really stable, and it helps the tertiary structure of the protein. But also an interesting thing of like, you can ask yourselves, oh, well, there's all these hydrophobic interactions, or this, like a protein could typically have infinitely many tertiary structures. So how does it get that conformation? Well, there's something called the hydrophobic effect, or how does this happen spontaneously? If there's something called the hydrophobic effect, and proteins fold the way they do because they want to maximize the entropy increase. So the delta S, I'll change the color of that. They want to maximize the delta S of water surrounding it. So this is just a brief overview of the hydrophobic effect. I can talk more about it in a future video, but I'm just kind of giving you guys a brief overview of just levels of protein structure. And a huge example of this that has tertiary structure, well, all proteins have tertiary structure, but we're going to look at, in particular, myoglobin, which if you didn't know, it's a protein that's in your tissues. It's responsible for storage of oxygen. But that's the overview of tertiary structure. So let's get into quaternary structure. Now, quaternary structure is interactions between two or more polypeptides. So this is like when you have tertiary structure of one protein and tertiary structure of another, and they come together to form that quaternary structure. A big example of this is going to be your hemoglobin molecule, because it has four subunits. So this is subunit 1, 2, 3, 4. And I definitely didn't draw this right, but and you have the heme group in the middle. So this is an example of quaternary structure because you have four subunits. And another example of this is going to be like antibodies, because you have the two regions that come together and are linked by disulfide bridges. But again, that's kind of beyond the scope of this video. We can talk about it more later. But anyways, it's just a brief overview of the levels of protein structure. You have the most basic, which is the amino acid sequence, which is the primary structure. It's a peptide bond formed through dehydration reactions. So that's your most basic level. Then you have the secondary structure, which is your intramolecular hydrogen bonds. And that's how you get your alpha helixes and your beta pleated sheets. And the way to kind of alter that, or kind of stop that alpha helix from forming, is with the proline amino acid, which is going to be found kind of at the end of the alpha helixes. Then we get into tertiary structure, which that's going to be your hydrophobic interactions. That's kind of how overall the 3D structure of the protein forms. So this is a little bit beyond the secondary structure, which the secondary structure is like the local 3D configuration, whereas the tertiary is kind of like the overall configuration. And a big player in how the protein folds is going to be the hydrophobic effect. You're going to want to increase the entropy of water molecules around it. which is gonna make the reaction favorable, which that's why proteins fold the way they do. And a big example of the tertiary structure is the disulfide bridge, which is formed by the oxidation of cysteine molecules R groups, which is, as you can see here as I've illustrated. But again, we can get into that in another video if needed. Then we have the quaternary structure, which is basically interactions between two or more polypeptides. And an example of this would be hemoglobin, which hemoglobin is responsible for transporting O2 from lungs to the tissues and CO2 from the tissues to the lungs, which we can get into that in another video as well. Again, this is just a brief overview. But anyways, I hope this kind of was helpful. Good resource to watch. This is Attorney to Our Tutor. Thank you for watching. Please subscribe and let me know what you want me to do in the future. Also, I'd be willing to help solve some problems. If you want to email me or DM me some of your chemistry questions or biology homework questions, I'd be more than happy to try and solve them and make a video on it. Anyways,